play with a couple of ideas.
We always start.
Yeah. I thought I had one other, but I can't remember what it might have been now. So we'll just move forward, and I'll turn it over to Dave Wake, who's going to introduce today's speaker.
It's my pleasure to introduce Jing from Kunming, China. Jing is no stranger to Berkeley. She came here as a postdoctoral student originally, before she set up her lab in Kunming. Jing is from northern China, but she went to Chengdu for her PhD, where she got her PhD at Sichuan University. And she was a student of the famous Zhao Ermi, who was the leading Chinese herpetologist. She tells me she went to Chengdu specifically to study with him. And he, in turn, is not a stranger at Berkeley, because he was a Miller visiting professor in about 1990. He spent a full semester here. And as a result of that, my first student from China, Anming Tan, came from Chengdu and finished his PhD here in the 1990s. Zhao Ermi is the continuation of a very distinguished lineage of Chinese herpetologists that was established by an American biologist, Alice Boring, who moved to China in 1918. And she spent the rest of her career there. And she had many outstanding students, but certainly the most outstanding was C.C. Liu, who got his PhD at Cornell in 1934 and went back to China to establish this school, of which Zhao Ermi was in the direct series because he was C.C. Liu's student. Alice Boring wrote her book, Amphibians of China, in a Japanese concentration camp during the Second World War. That was published in 1945. C.C. Liu moved from northwestern China, wasn't he? Or northeastern China. And he moved to Chengdu to escape the Japanese invasion of China during the war. And that's how herpetology in China came to be centered in Chengdu, which is, it still is a very important center. The Academy of Sciences, the Institute of Biology there, is one of the two really important centers. And the other is now the Kunming Institute of Zoology, where Jing is now a professor with her own very active and productive laboratory. And she's spending the year here in the United States. She's centered in Austin, Texas, where she's working with David Hillis. And she's going to return there tomorrow. So I hope you have a nice rest of the year here, and we're looking forward to your talk on Qinghai-Tibetan Plateau.
Well, thank you so much, David, for the nice introduction. That's the history, or Chinese herpetology, how our relationship with the United States. So today, really, it's my great honor to be here to come back. I'm very happy to visit. So very happy to give this talk today. Take the opportunity, I would like to firstly briefly introduce my institute, Kunming Institute of Zoology, Chinese Academy of Sciences. So the Chinese Academy of Sciences is quite different from the American Academy of Sciences. So Chinese Academy of Sciences comprises over 100 institutes across China, along with 3 universities, so involving multiple fields, research fields. So Kunming Institute of Zoology is one of the institutes on the biology, located in the beautiful city, Kunming. Kunming Institute of Zoology is located just here. So it's located near the Indo-Burma Biodiversity Hotspot. Lies in the here is the southwestern part of China, Yunnan Province. Yunnan Province is the biodiversity kingdom in China. Our institute is one of the research center on biodiversity, conservation and utilization of animal resources. My lab is one of the 36 research group in this institute. Certainly we have other several main research priorities. Very strong team, several groups works on the evolutionary genomics. My lab, the interest, our interest generally quite broad, but still focused. Overall, we want to explore and understand the pattern and mechanisms forming the faunal diversity. We focus on the amphibian reptiles. Personally, me more amphibians, but my students begin to work on reptiles, so we move together. We work together. This involves phylogeny, biogeography, evolutionary biology. Of course, for the responsibility, we do some work on conservation in China. The regions we are interested in generally in China and Asia, especially in Southeast Asia. Since many species, Chinese species groups, is not Chinese species groups, they are not the monophyletic on the tree of life. So we have the close, the species have the close relationship with the other belt across the border. Certainly they have no the visa problem as us. So we have developed broad international cooperative survey in recent years, and we aim to pursue to better understand the using an evolutionary way to understand the Chinese herpetofauna, especially on the tree of life. In the lab, we explore the diversity. We describe new species. This is very basic, but quite important for our further study. Based on the base work, so we, to a higher level, we often pursue the pattern and process of biodiversity forming and test the hypothesis of speciation. Beyond this, we also ask the question on the genetic adaptations, how these frogs and amphibian reptiles live on, cope with the extreme environment. Today, my talk mainly focus on the project on the Qinghai-Tibetan Plateau. As we know, the biodiversity is unevenly distributed on Earth. We often ask how and why biodiversity varies and evolves in time and space. Why some places higher diversity occur there, not in other regions. We scientists often have the particular interest on the origin and the evolution of biodiversity in those mountain regions. The Qinghai-Tibetan Plateau is one of the model regions for us to explore this kind of question. I think, I trust for many of us, the Qinghai-Tibetan Plateau is unusual, is enigmatic, and it's the highest and largest plateau in the world. Geologically, the uplift of Qinghai-Tibetan Plateau resulted from the collision of the Indian plate with the Euro area, happened around 55 million years ago. this clearing definitely has changed the topography of area and caused the global climate changes, including the Asian monsoon system, development of the Asian monsoon system. The geological history is quite complex, but it has caused the first-order geological features. Sometimes we term the Qinghai-Tibetan Plateau as the third pole because here has the largest volume of ice and snow outside the polar region. So in recent years, with the global climate changes, there are intense interest on how the climate changes would cause the changes in this region. And you see here, it's the world's most important water tower. It has been the source of 10 largest rivers in Asia. From this picture, you can see Qinghai-Tibetan Plateau is a vast elevated plateau surrounded by many mountain ranges. The famous Mount Everest is the tallest peak of the world, occurs in the middle part of Himalayas. The K2 is the second tallest peak of the world, occurs in the western part of Himalayas. Of course, there hosts exceptional diversity and a very high level of endemism. It's the home to the unique flora and fauna. In the fringe of the Qinghai-Tibetan Plateau, there are 4 biodiversity hotspots, including mountains of Central Asia, Himalayas, and Indo-Burma, and the mountains of southwest China. So our study focuses on this region. Our general question is, how did the species evolve here? Compare, I know many people here work on Andes. So compared to the study in Andes, so the evolution of wild in the Qinghai-Tibetan Plateau generally remains poorly understood, despite its outstanding, geological extent. So I use the amphibian reptile as a model system to explore this question. So amphibian reptiles, they are sensitive to the environmental and climate changes. Generally, they have poor dispersal ability compared to the birds and the plants, other organisms. Today, my talk mainly focuses on below the 3 parts. The third part is on the species richness and patterns of the herpetofauna in the Tibet autonomous region. It's part of the Qinghai-Tibetan Plateau. There has been limited documentation of herpetofauna study in this region, mainly because much of this region is previously not accessible or very hard to enter. Even today, for example, the high elevation dead zone. So some parts of Himalaya region is still with the primary forest there. To better understand the diversity there, since 2010, we have organized the very thorough field survey in this region. A total of 23 trips involving 26 teams. We visited 251 localities in this region. Many new taxa previously in recent years discovered and described by my group, including new family records of Ceratobifurcated. Previously, this family only documented in the southeastern Asia, the island of Southeast Asia. Our study has expanded its distribution range to the Himalayas, and definitely to the northernmost and to the westernmost. We published the new genus here along the Himalayas, and 20 new species published in recent years. With our efforts, we document 60 amphibians here and 79 reptiles. Compared with the species numbers based on the first Qinghai-Tibetan scientific expedition, our study documents 23 additional species of amphibians and 26 more reptiles. The first Qinghai-Tibetan scientific expedition was organized by the Chinese Academy of Sciences in 1970s. Many senior herpetologists in China joined that expedition, including my supervisor, Ermi Zhao. He was one of the persons. Unfortunately, he already passed away 3 years ago. Based on the collection during the expedition, the famous Chinese woman herpetologist Shu Qinhu, he finally wrote the book and published the data out. Based on our analysis, very interestingly, we found that this region, its Yaluzangpu Grand Canyon region, has the richest diversity, up to 45 amphibians and 43 reptiles, belong to 7 families and 9 families respectively. This region, the Grand Canyon region, is the largest canyon of the world, as I know. Here is the Mount Nanjabawa. So Yaluzangpu, this gorge is bent around this mountain. It's very high elevation. So bends around Mount Nanjabawa, and then cut its way through the eastern part of Himalaya. Finally flow into India. It becomes the Brahmaputra of India. So very unique ecosystem, and with many endemic species here. And you see here the vegetational zone shows very distinct elevational gradient. The climate ranges from the tropical to the Arctic, provides a variable environment for amphibian reptiles, actually. Of the 17 families in Tibet, among these 7 families exclusively occur in this Yaluzangpu Grand Canyon region. I highlight here in the red color. I take an example to introduce my favorite frog. We call tree frog, but it's quite different from your tree frog here. Your tree frog is holiday, so this is only in the older world. Definitely here is a wonderland for the tree frogs. This region has 8 genera and 13 species. So we have 5 species of this rock forest, this genus. Very beautiful frog. And we have 2 species of Xylodema. So it's very hard to get them. Even you hear the sound in the forest, it's very hard to get them. The first one we often call is bird-shaped frog. This one, we have 1 polypedis, 1 rochestis, also very tiny frog. And one Cruexellus, one Fihela, one Gracioxellus. My favorite frog is this one. Nasuteaxellus montoensis. Actually, my group described this new species, and we established this new genus. Currently, this genus only occur along the Himalayas. In China, we have 2 species, and the third species in India. This one locality of Xigong Lake in the Yaruzangbu Grand Canyon region. Around the lake, you could find a total of 6 general and 8 tree frogs co-occur here. It's still no way to enter, so you need to, from the village, you need to climb 2 mountains to enter. So it's in the during the raining season, it's very dangerous, flooded. So generally, back to the whole Tibet, based on our analysis, generally the diversity mostly occurs in the southern Tibet and the eastern Tibet. It's the Himalaya region and the Hongduan mountain range. So how can we understand this remarkable diversity in those mountain regions? So the second part, I would like to introduce one of our recent project on the origin and diversification of bird fauna along the Himalayas. Regarding the uplift of Himalayas, there are generally 3 hypotheses could be concluded. The first one is early original hypothesis. This is a model in which the Himalayas arose during the Paleogene. This model mostly are supported by the earlier studies. The second hypothesis we view as the intermediate stepwise hypothesis. It's a model the Himalayas arose in the early Paleogene, but with an elevated rate during the Miocene. The third one is we call late original hypothesis. It's a hypothesis. This is a model in which the Himalayas arose generally quite late after the 50 million years ago. So if the faunal assembly tracks the if the faunal assembly tracks the geological history, we would expect the similar path of the uplift-driven diversification of belt, including in-situ diversification and the dispersal between different regions. This is a popular hypothesis, but little tested. Very interestingly, in recent years, one biogeographic study on the 19 Himalaya plateau. So their study shows the diversification dynamics of the flora in the Himalayas initially quite stable during the time. Here you could see. So their study suggested a possible gradual uplift of Himalayas during the early to middle Miocene. Regarding the origin of Himalaya belt or fauna, so generally it had been generally accepted that it consists of elements from the adjacent region. For example, from West Asia, from East Asia, from Southeast Asia. So we view it as the sink hypothesis. So most current biogeographical studies on this region have been focused. They often use the endemic species groups and often, with high dispersal ability, including birds and plants, butterflies. So certainly it's good, but if you use the endemic species groups, often without a global perspective, the dispersals between different regions is often overlooked, and there are very few studies have used the comparative cross-taxa analysis. So the general contribution between institute diversification versus the dispersal between different ranges is not clear. So we use the amphibian reptile as model. We want to test those specific hypotheses, scenarios of geological uplift and the uplift-driven diversification of belt, and also the origin of Himalaya fauna. Our dataset for the phylogenetic analysis finally includes 14 families and 85 genera involving 1,628 in-group species. Among these data, 223 species occur in this region along Himalayas. Finally, we choose 37 clades for the biogeographic analysis, and based on the results, we calculated the two events, including in-situ diversification and dispersal, related to Himalayas. Totally, there are 285 biogeographic events. Very interestingly, you could see here there are significantly higher accumulation events in this region, in this time range, in the Miocene, over 60% in this time range. So among these events, in-situ diversification events contribute more than half. Here is the overall look. You will see in-situ diversification and dispersal generally shows a similar increased trend. The dispersal events slightly lagged behind of the in-situ diversification. But both of them have the increased rates during the early Miocene to the middle Miocene. And both of them have the peak hit around 13 million years. So clearly our analysis cross-taxa amphibian reptiles, our study support the geological hypothesis B is intermediate stepwise origin. It's quite different from the Aurora study. If we take a closer look at the interchanges between Himalaya and the adjacent regions, including Himalaya and Southeast Asia, and Himalaya and East Asia, Himalaya and the South Asia and the Western Asia. So generally, clearly, Southeast Asia has the most interchanges with the Himalayas, totally occupy over 90% of all the dispersal events. For all the dispersal events, dispersal into is significantly higher than the dispersal out of the Himalaya. So our study found both in-situ diversification and dispersal have important roles, but in-situ diversification have predominated, and our study just based on our study, a sole sink hypothesis is rejected. Itself cannot explain the origin of the herpetofauna in this region. A quick summary here. Using comparative cross-taxa analysis, our study found Himalaya herpetofauna has a rapid increase of diversification during early to middle Miocene, which support one of the geological scenarios recently proposed by Ding et al. 2017. Our analysis is the first to find a predominant role of in-situ diversification over dispersal in the assembly of Himalaya belt. The third part, I would like to move to the high elevation adaptation. Last week, I joined the MVZ symposium, so I heard Michael's lab also have some this camera. I'm very interested. As we know, it's quite challenging to live on the high elevation. So it's oxygen is definitely scarce. Very dangerous. UV radiation, very dangerous high, and the temperature is quite low there. So the endemic species in this region face challenges few other lowland species experience. But we have realized the high elevation is not a single part, but represents a continent. Generally, with the increase in altitude, there is a percentage of decrease of air and oxygen pressure. So my point here is the high elevation pressure is variable along the elevation gradient. So our question is, what elevation did the high elevation adaptation first emerge at increasing elevations? Did the elevation of high elevation adaptation follow a continuous process, or just happened on a specific or particular elevation threshold? And is there any evolutionary convergence between different groups? That's our major three questions. To answer these questions, it requires a model along an elevational gradient. In recent years, there has intense interest on the high elevation adaptation, but most of previous studies have focused on comparisons between pairs of high elevation versus low elevation species. That's actually good. They, in recent years, published a lot of papers, and they got some very interesting findings. But this kind of project design cannot answer all questions. So we are lucky. We have two excellent groups. One is amphibian nanorana. Another one is reptile, is Phrenocephalus. The two species groups distribute through the interior plateau to the adjacent lowland. Based on the well-resolved phylogenetic relationship, finally we choose 4 in-group representative species for our study in this study. We did the same thing for the sand lizard. In this study, we used the RNA sequence data, and we collected multiple tissues, and we pulled them together, and finally we generated de novo transcriptomic assembly for each species. By this way, we could to minimize the gene numbers we could examine in this study. So finally, based on the super gene elements, we built the time tree. We use different colors here. We use different colors to label those lineages at different elevations, and we estimated the selection pressures along each lineages. Here is the red color is the highest. Those represent those lineages as very high elevations. Black color is lowland lineage. And we did the comparative evolutional analysis. In the first stage, we identified a quite high number of positive selection genes along the green lineages compared with the lowland lineages. Many of these positive selection genes can be enriched into below those functional categories. Each of these functional categories is closely related to those physiologically stress produced by the UV radiation and hypoxia. So this analysis strongly supports both frogs and lizards have evolved molecular adaptation in the earliest stage around 2,000 meters. When we move to the higher elevation lineages, we still can find a high number of positive selection genes, including 3,500 meters and even the very high elevation, about 4,500 meters. So very interestingly, if we take a look, all the positive selection genes enrichment date at increasing elevation, we found those genes related to DNA repair and energy metabolism had experienced rapid evolution. This indicates DNA repair, energy metabolism. They are the two important classes, at least for frogs and lizards, for them to cope with the high level of adaptation stress. So even the two species groups are quite distantly related, but we still want to ask if there are convergent evolution. The answer is yes, but you see here, at each comparably elevations, we found a number of functional categories have evolved convergently in both of the group, both frog and lizards. But if we take a closer look at each comparable elevations, very few genes are shared by both of them. So this indicates many of the convergent functional actually are associated with different set of positive selection genes. So if we consider all the positive selection genes between the two groups, we can find a total of 32 parallel positive selection genes identified. Indicated later they could be the target genes to the functional experiments. So a quick summary here. By studying species groups across elevational gradients, our study found the high elevation adaptation is evident in the earliest stage at around 2,000 meters. We found the continuous evolution of high elevation adaptation with increasing elevations. Between the two species groups, there are convergent evolution, but more at a functional level, not the specific gene level. Recently, we have reviewed publications from 12 amphibian reptiles occurring on this region. Generally, a network of pathways of gene under positive selection gene can be concluded here. It's still very few genes are convergently evolved by more than 3 species, only one here, you see, shared by 3 lizards. But most of this, all these positive selection genes can be classified into 3 major functional classes, including DNA repair, hypoxia, and metabolism, indicating their important role for the amphibian reptile to cope with the high level adaptation, to cope with the high level stress. My lab is still working on this topic, high level adaptation, but we know it's complex traits actually, and many systems involved. And currently, one of the scientific questions I'm quite interested is on how these frogs on the plateau to respond to the strong UV radiation. If you work on Tibet, this is I have studied this species for a long time. You cannot believe it could live up to 5,000 meters. Very strong UV radiation there. It's quite particularly important for frogs. Their skin is bare. So with the global anthropogenic climate change, there are increased UV radiation due to the ozone depletion. So if when this happened one day, I mean, this already happened. So how this frog cope with this stress? Is there the frog have this ability? So I'm interested in these questions. So we already developed the system recently in the lab. Compared the lowland species, Qinghai-Tibetan Plateau, we found the Tibetan endemic frog, Nanorana parkeri, had developed effective free radical scavenging ability. You can see here the Nanorana parkeri. Under the UV exposure experiment in the lab, so you can see Nanorana parkeri can very quickly to scavenge the free radical just around 30 minutes to 100%. But for the lowland species, Qinghai-Tibetan Plateau, even with longer time, still it's just 20%. It's quite low. So one of my students now is working on the collected phenotypic data and the genotypic data. So I hope later we have a chance to communicate on this part. Certainly, the lab still working a lot on the large-scale diversification of cropped fauna in this region, and I'm interested on the speciation is to test those hypotheses of speciation in this region. So overall here I want to see the Qinghai-Tibetan Plateau is a natural history for studying diversification and evolution of wild. And the amphibian reptile definitely is one of the ideal models to explore below questions, including how environment influence the evolutional history of species here, how selection drives adaptation, and sometimes how speciation can occur, how species cope with the high level stress. For this part, today I have no time to talk about more, but definitely my lab is still working on that, but mostly by population genomics work. So hope later, especially, we would have time to talk about this part. So generally, I would like to see all the studies here on the Qinghai-Tibetan Plateau may help guide priority in conservation efforts cross both regional or species level and facilitate forecasts about ecosystem service and future climate change. What I talked today is part of the contents in this book, Amphibians and Reptiles in Tibet. It will be published this year, so I hope it will come out this semester. In the end, I would like to thank for people here. Yating Zhang is my colleagues in my institute. David Hillis, David Wick here, Robert Murphy from RAM. So we have continuously worked together on those projects on the Qinghai-Tibetan Plateau for many years. Here I would like to thank all the 10 persons. They did specific work, including through the field, very hard field survey to the genomic evolution work. And I have many collaborators in China and work together on different projects on the Qinghai-Tibetan Plateau. And Forest Department of Tibet issued the permit for us and give us much help. And this indeed the efforts from the whole lab. And in the past years, I'm lucky to get some funding from China, especially from Chinese Dynamic Sciences. Finally, I would like to specifically thank David Wick and MVZ. I got training 10 years ago here in his lab and have known many people. So David definitely is a top scientist on salamander amphibians. What he did actually has inspired me and encouraged me to take brave steps to pursue my passion on herpetology in China. So I'm very grateful. And also, people here have been so nice in MVZ, so I always would love to come back. I saw many friends, many people still here. I know them since 10 years ago. So here I wish MVZ everyone here a wonderful life and successful research. And I joined the symposium, so I enjoyed the talk. So Happy New Year!
If somebody could get the lights there at the door, we have time for questions.
I would be happy if you have any questions or other questions after the talk.
I have a question. So, in your analysis indicating or finding the genes under selection on the different branches, do some genes pop up several times on the different branches? Do you see like increasing levels of selection?
You mean across different elevation levels?
Yeah.
I mentioned very few. Those genes actually, all the genes cross. You mean?
So would if one gene appear to be positively selected
In
on more than
different
one branch?
lineages? You can see here it's very few.
Yeah. I'll have to read
So that's why. So we feel originally we think there should be have some convergent, gene, convergent among different lineages. But finally, it's kind of that's way we think is a kind of functional category convergent, but not specific genes we found.
How do you do it? It's a follow-up to this, so let me go ahead and ask it. So yeah, I was really curious about this, the 8 or 10 genes that you identified that were convergent in the 2 lineages. And because you didn't really spend much time talking about those. And I'm wondering if there are any like particularly compelling genes there that turned out to be positively selected in the frog and in Phytocephalus.
You mean between the 2
Yeah,
groups?
like I think your next slide or maybe the previous one, you indicate that there are, here, so there are 6, 2, and 1 at the different elevations. Like that one gene that's shown to be
Yeah,
strong
very
and
high, very high.
very high,
elevation,
both
yeah.
in frogs and in the
Yes.
Phytocephalus. Is that gene something is that particular gene a particularly interesting gene like HIF or a hemoglobin gene or something where you'd really expect it to be under
Yeah.
strong selection?
Not expected actually. We found that gene, but some genes I think we don't know the function even.
Ah, okay.
You know.
Yeah.
Yeah.
I know it's very difficult to get complete genomic sequences for frogs, but you've been successful with Nanarana parkeri. And do you expect to continue that effort, or are you going to be happy with the methods you're using now? Do you want to go to whole genomes, or
For which question? I mean,
for
for the
these-
frogs?
Yes.
Nanarana parkeri.
Yeah. Well, for
Yeah,
more
right.
frogs.
For more frogs.
Yes.
That's hard,
Or is it just not worth the effort?
Depend on which kind of question we will ask.
Yeah.
Then I will sequence that frogs. But generally, I think I'm lucky because this frog only 1.3
Yes,
gigabytes.
it's
So
small.
if a larger one, it's still hard. I think.
Yes.
So these species span a big range of elevations,
Yeah.
right? What do you know about the acclimation response? If you take a frog from very low elevation, one of these that spans, and bring it up to very high elevation, have you measured phenotypes that vary as a result of acclimation to a different
One of my students is working on that. We did a little bit on the Tibet, take the species to the higher. Currently I have noticed it out. Yeah, we did that actually.
Right, and what phenotypes are you looking at when you do that?
Including the hemoglobin, or as I remember, I discussed this hemoglobin changes with my students, but I don't think he works this out.
So like, I'm just kind of curious about the frog lineages. Like you mentioned that have greater ability to respond to ultraviolet light. Like, does that sort of imply that those lineages invading that region earlier, or?
You mean the
Yes,
this
like
frog?
yes, like you mentioned that those lineage have better ability to respond to UV light.
Yeah.
So like yeah, I'm just curious if that implies that they invaded in that region earlier than other lineages of frogs.
No, this is quite. This is a time. Actually, it's a time tree candle. So
Oh, okay.
yeah, that's
So-
much younger compared to this one. This is about 23 million years ago. But this one, I think Nanarana went to the Tibet just about 8 million years.
Okay.
Okay, if there are no more questions, let's thank Jing again.